Nucleic acid is a polymeric macromolecules made up of a repeated unit of monomeric nucleotides. We have two main types of nucleic acid, that is the deoxyribonucleic acid and ribonucleic acid, or what we call as DNA and RNA. Based on the structure, we can differentiate these two because DNA is a double helix while RNA is a single. Both DNA and RNA are made up from nucleotides. So, what is nucleotides? Nucleotides are the unit structure of nucleic acid, composed of three components: the nitrogenous base, pentose sugar, and phosphate. As you can see in the illustration, we have nitrogenous base in the center, thymine paired with adenine, guanine is paired with cytosine, sugar and phosphate that serve as the backbone. One of the components of nucleotides is the nitrogenous base. We have two types, the purine and pyrimidine. For purine, it is a two-ring structure. We have adenine and guanine. For pyrimidine, it is a single ring structure. We have cytosine, thymine, and uracil. In this table, we have the nitrogenous base, nucleoside, and deoxynucleoside. For the base, we have adenine, guanine, cytosine, uracil, and thymine. Nucleosides of this nitrogenous base: for adenine, we have adenosine. Guanine is guanonosine. For cytosine is cytidine. Uracil is uridine. For thymine is not usually found. And for these deoxynucleosides, for this nitrogenous base, we have 2-deoxyadenosine for adenine, 2-deoxyguanonosine for guanine, 2-deoxycytidine for cytosine, and 2-deoxythymidine for thymine. Deoxynucleosides for uracil is not usually found because uracil is nitrogen base of RNA or ribonucleic acid. That's why it indicates that it's not usually found in this table. We have two main types of nucleic acid. We have deoxyribonucleic acid, or what we call DNA, and the ribonucleic acid, or what we call RNA. For pentose sugar of DNA, we have deoxyribose. Oxygen is not present in position 2, while the pentose sugar of RNA is ribose. Oxygen is already present in position 2. For the nitrogenous base of DNA, for purine, we have adenine and guanine. For pyrimidine, we have cytosine and thymine. And for nitrogenous base of RNA, we have adenine and guanine for purine, and cytosine and uracil for pyrimidine. Thymine is replaced with uracil in the phase of RNA.
Ribonucleic acid. Pentose sugar is ribose. Bases are purines A and G, and pyrimidines C and G. Linear polymerization of nucleotides. Nucleic acids are formed of nucleotide polymers. Nucleotides polymerize together by phosphodiester bonds via condensation reaction. The phosphodiester bond is formed between hydroxyl OH group of the sugar of one nucleotide and phosphate group of other nucleotide. As seen in the figure below, the bases are interconnected via a phosphodiester bridge, which is composed of the occurrence when exactly two of the hydroxyl groups on each different molecules to form two ester bonds, and by then eliminates H2O, or the water molecule, thereby providing structural support for a chain of bases. Attached to each sugar is a pair of two bases, adenine partnered with thymine, and cytosine partnered with guanine. The two strands are held together by hydrogen bonds between the bases, with adenine forming a base pair with thymine, and cytosine forming a base pair with guanine. Significance of complementary base pairing. The importance of such complementary base pairing is that each strand of DNA can act as a template to direct the synthesis of other strands similar to its complementary one. Thus, nucleic acids are uniquely capable of directing their own self-replication. The information carried by DNA and RNA direct the synthesis of specific proteins which control most cellular activities. The DNA structure. DNA is a double-stranded molecule consists of two polynucleotide chains running in opposite directions. Both strands are complementary to each other. The bases are on the inside of the molecule, and the two chains are joined together by double H bond between A and T and triple H bond between C and G. The base pairing is very specific, which makes the two strands complementary to each other. So each strand contains all the required information for synthesis replication of a new copy of its complementary. This is vital for the cell for it to manufacture new cells with the same DNA structure to perform identical tasks. The formed polynucleotide chain is formed of negative charge sugar phosphate backbone, free 5-phosphate on one end, free 3-hydroxyl on other end. Nitrogenous bases are not in the backbone. Attached to the backbone, free to pair with nitrogenous bases of other polynucleotide chain. Nucleic acids are polymers of nucleotides. The nucleotides formed of purine or pyrimidine bases linked to phosphorylated sugars. The bases are linked to the pentose sugar to form nucleoside. The nucleotides contain one phosphate group linked to the 5-carbon of the nucleoside. Nucleotide is equals to nucleoside plus phosphate group. The polymerization of nucleotides to form nucleic acids occur by condensation reaction by making phosphodiester bond within 5-phosphate group of one nucleotide and 3-hydroxyl group of one another nucleotide. Polynucleotide chains are always synthesized in the 5-to-3 direction, with a free nucleotide being added to the 3-OH group of a growing chain. Complementary base pairing. It is the most important structural feature of nucleic acids. It connects bases of one polynucleotide chain with complementary bases of other chain. Complementary bases are bonded together via double hydrogen bond between A and T and A and U, and for RNA, A and T or A and U. Triple H hydrogen bonds between G and C in both DNA or RNA, G and C. 5-to-3 polynucleotide linkages. Phosphodiester bonds 5 and 3 links to pentose sugar. N glycosidic bonds links nitrogenous bases to C1, pentose, and beta configuration. The essential features of bDNA. Right twisting double-stranded helix, antiparallel, base on the inside perpendicular to axis, uniform diameter, major and minor groove, complementary base pairing. The forms of DNA. The first one is B-form helix. This is the most common form of DNA in cells. Right-handed helix turns every 3.4 nanometers. Each turn contains 10 base pairs. The distance between each two successive bases is 0.34 nanometers. Contains two grooves. Major groove, or wide, provide easy access to bases. Minor groove, narrow, provide poor access. The next one is A-form DNA. This less common form of DNA, more common in RNA. Right-handed helix, each turn contain 11 BP per turn. Contain two different grooves. One is the major groove, it is very deep and narrow, and lastly minor groove, very shallow and wide, binding site for RNA. The last one is Z form of DNA. Radical change of B form. Left-handed helix, very extended. It is GC-rich DNA regions. Sugar-based backbone form zigzag shape. The B to Z transition of DNA molecule play a role in gene regulation. In nucleotides, phosphate nucleotide monomer ester, monomer of pentose dinucleotide or dimer. Oligonucleotide are short polymer. Polynucleotide are long polymer. And nucleoside are monomer of sugar plus base.
DNA is a more stable molecule than RNA, which is useful for a molecule which has the task of keeping genetic information safe. RNA containing a ribose sugar is more reactive than DNA and is not able in alkaline condition. To be able to understand the DNA, let us know the various forms of the DNA or the DNA conformation. Different forms of the DNA can be formed depending on its interaction with surrounding condition. First is the B form. B form has intermediate shape with wide and quiet deep major groove and narrow and quiet deep minor groove. It is a right-handed double helix. It has a high relative humidity, form approximately 92%, which is an ideal environment of a cell. B form is the most common form inside the cell. A has a broadest shape with narrow and very deep major groove and very broad and shallow minor groove. It is also a right-handed double helix. It is represented in a dehydrating environment with humidity of 75% and higher concentration of ions. B form can take a form in order for the cell to survive. Z. It has a narrow shape with flat major groove and very narrow and deep minor groove. It is a left-handed alternating helix. Z form is named Z since it is a zigzag form. I am Benladin Guiamad, Section BSN 1A.
RNA is generally single-stranded intramolecular H-bond base pairing occur between complementary bases on the same molecule or secondary structure. The petose sugar is ribose, uracil or U replace thymine or T in the pyrimidine bases. The secondary structure of RNA consists of single polynucleotide. Base pairing in RNA occurs when RNA falls between complementary regions. Both single and double-stranded regions are often found in RNA molecule. Types of RNA. First, messenger RNA or mRNA. mRNA carries genetic information copied from DNA in the form of a series of 3 base code, each of which specifies a particular amino acid. Second, transfer RNA or tRNA. tRNA is the key that reads the code on the mRNA. Each amino acid has its own tRNA, which binds to it and carries it to the growing end of polypeptide chain. Next, ribosomal RNA or rRNA. rRNA associated with a set of proteins to form the ribosome. This complex structure, which physically moves along the mRNA molecule, catalyzes the assembly of amino acid into protein chain. They also binds tRNA that have the specific amino acids according to the code. It can take three levels of structure. First, the primary sequence of nucleotides. The primary structure denotes the ribonucleotide sequence, commonly referred to as a base sequence of the molecule. Usually, the base sequence of RNA molecule only consists of a combination of the bases adenine, guanine, cytosine, and uracil. Thymine and hypoxanthine are analogous to uracil or adenine respectively and thus represented by their corresponding standard base. Secondary hairpin loops or base pairing. The secondary structure is formed by a subset of the Watson's Crick base pairs contained in RNA molecule. This includes the standard canonical adenine and uracil and guanine and cytosine. Pairings already known from the formation of DNA helices as well as guanine and uracil's Weber pairs. Successive base pairs forms energetically variable and thus stable stem regions. Tertiary motifs and redefolding. Tertiary interactions can occur between two helical motifs or stem-to-stem, between two unpaired regions, loop-to-loop, and between an unpaired region and stem region, or loop-to-stem. Transfer RNA or tRNA structure is a type of RNA molecules that helps decode a messenger RNA or mRNA sequence into a protein. tRNAs function at specific site in the ribosome during translation, which is the process that synthesizes a protein from an mRNA molecule. Proteins are built from smaller units called amino acids, which are specified by three nucleotide mRNA sequence called codons. Each codon represents a particular amino acid, and each codon is recognized by specific tRNA. The tRNA molecule has distinctive folded structure with three hairpin loops that form the shape of a three-leafed clover. One of these hairpin loops contains a sequence called the anticodon, which can recognize and decode an mRNA. Each tRNA has its corresponding amino acid attached to its end. When a tRNA recognizes and binds to its corresponding codon in the ribosome, the tRNA transfers the appropriate amino acid to the end of growing amino acid chain. Then the tRNA and ribosome continue to decode the mRNA molecule until the entire sequence is translated into a protein. tRNA are one of the kind of RNA that help in protein synthesis by serving as the physical link between the mRNA and amino acid sequence of protein. They carry their amino acid toward the mRNA strand. They pair onto the mRNA by way of anticodon on the opposite side. Types of RNA. Messenger RNA or mRNA codes for proteins. Messenger RNAs are one of the types of RNA that are found in the cell. This particular one, like most RNAs, is made in nucleus and then exported to the cytoplasm, where the translation machinery, the machinery that actually makes proteins, bind to these mRNA molecules or reads the code on mRNA to make specific protein. So in general, the DNA for one gene can be transcribed into an mRNA molecule that will end up making one specific protein. Transfer RNA or tRNA adapter between mRNA and amino acid. Transfer RNA or tRNA is a type of RNA molecule that participates in protein synthesis. Each tRNA molecule has two important areas: a trinucleotide region called the anticodon and a region for attaching a specific amino acid. During translation, each time an amino acid is added to the growing chain, a tRNA molecule forms base pairs with its complementary sequence on the messenger RNA or mRNA. ensuring that the appropriate amino acid is inserted into a protein. Ribosomal ribonucleic acid, or rRNA, is the RNA component of ribosome, the molecular machines that catalyze protein synthesis. Ribosomal RNA, or rRNA, forms ribosome core for translation. Ribosomal RNA constitute over 60% of the ribosome by weight and are crucial for all its functions. From binding to mRNA and recruiting tRNA to catalyzing the formation of a peptide bond between two amino acids. Even the structure of ribosome is determined by the three-dimensional shape of its rRNA core. Proteins present in the ribosome serve to stabilize this structure through interaction with the core. Besides mRNA, tRNA, rRNA, eukaryotics have some RNAs. These are heterogeneous nuclear RNA and small nuclear RNA. Small nuclear RNA involved in post-transcriptional processing. Replication of DNA molecule is semi-conservative, which means that each parent strand serves as a template for new strand and that the two new DNA molecules each have an old and one new strand. DNA replication requires a strand of DNA to serve as a template. Substrate, deoxyribonucleoside triphosphate. DNA polymerase. DNA polymerase an enzyme that brings the substrate to the DNA strand template. And a source of chemical energy to drive this synthesis reaction. Nucleotides are always added to the growing strand at the 3' end. The hydroxyl group reacts with the phosphate group on the 5'C of the deoxyribose, so the chain grows. Energy is released when the bond linking two of the three phosphate groups to the deoxyribonucleoside triphosphate breaks. Remaining phosphate group becomes part of the sugar phosphate backbone. Step 1. Unwinding and exposing strands. Synthesis of new DNA strands proceed in both directions from an origin of replication, resulting in bubble with replication forks at each growing point. DNA strands are unwound and opened by enzymes called helicases. Helicases act at specific places called origins of replicates. Step 2. Priming the strand. A primer is a short single strand of RNA or ribonucleic acid, and it's complementary to the DNA's template strand. In order to begin making a new strand, a helper strand called a primer is needed. Primers are formed by enzymes called primases. Strand elongation. Two molecules of DNA polymerase III clump together at replication forks, each acting on one of the strands. One strand exposed at 3' and produces a daughter strand which is called the leading strand. This strand is synthesized continuously and grows from 5' to 3'. The second daughter strand is called the lagging strand and it's antiparallel to the leading strand. The lagging strand is constructed in small backward-directed bits consisting of discontinuous section of 100 to 200 nucleotides in eukaryotic and 1000 to 200 in prokaryotic, called Okazaki fragments. When an Okazaki fragment forms, DNA polymerase once removes the RNA primer and replaces it with DNA adjacent to the fragment, leaving one bond between adjacent fragments missing. A second enzyme called DNA ligase catalyzes the formation of final bond. This is how DNA end up in the nucleus. Telomerase is reverse transcriptase that adds nucleotides to the 3' end of lagging strand DNA molecule. Telomerase prevents shortening of lagging strands during replication of linear DNA molecules such as those of eukaryotic chromosomes.